Hello students, I welcome you all at Infinity Learn by Sri Chaitanya. My name is Kiran Chaudhary and I am your biology faculty. Students, today I am back with another interesting topic, that is pre-fertilization structures. Yes, this is which chapter related to which chapter? It is chapter number 2 in your 12th class NCERT biology textbooks. The chapter name is Sexual Reproduction in Flowering Plants, right? So, the topic that we will discuss today in today's session, right, is related to pre-fertilization structures, and the structures that I am going to discuss with you today are Stamen and Microsporangium. These are the two structures we need to understand in detail, right? So, before jumping into the details of today's session, I would love to give you a brief overlook of what this chapter is about, right? So, this thing, the stamen and microsporangium, is covered under which topic? Pre-fertilization structures. Now see here students, why are we studying the pre-fertilization structure? This is very important. You got to understand what is the objective of understanding these points, right? The stamen and microsporangium. Why are we doing it? So listen very carefully students. The objective of this chapter, the chapter name is Sexual Reproduction in Angiosperms or Flowering Plants, right? So the objective of this chapter is to form what? Or to form a zygote, to form a seed, right? That will be done. This is result of sexual reproduction, right, in a flowering plant. Now, to do sexual reproduction in flowering plants, what is the prerequisite conditions? You need to have male gamete and female gamete, right? So, when male and female gametes are produced inside a flowering plant, only then sexual reproduction can take place, right? So that is the objective of this entire chapter. Now, to get to that stage, right, where fertilization can take place, we need to understand how these male and female gametes are formed, right? So in today's session, right, what are we going to study? That exactly where, yes, exactly where these male gametes are formed. So that's the objective. So let me tell you in advance that the male gametes, today we will talk only about male gametes, not the female one, right? So male gametes are actually produced inside pollen grain, right? So to get to that male gamete, we need to understand where exactly pollen grains are formed, right students? So from here we will start today's session. We will try to find out inside a flower where pollen grains are formed, which are ultimately responsible for the formation of male gametes, right? So let us start the session today. So here on the board, what you can see is a beautiful image of a flower, right? What you can see is this, a globular, sorry, I need to take pen first, 1 second. So what is this? This portion students, this globular portion and this filament वाला portion. So what is this? तो ये दोनों मिला के आपको जो दिख रहा है वो क्या है? वो क्या है? वो मेल रिप्रोडक्टिव ऑर्गन्स है फ्लावर के, राइट? यानी कि क्या है? स्टेमन है. So see here students, this what are we going to study is this. तो आज के हमारे लेक्चर का ऑब्जेक्टिव है इस पार्ट को स्टडी करना, क्योंकि इसी के अंदर बनेंगे पोलन ग्रेन्स और पोलन ग्रेन से बनेंगे मेल gametes. तो here we have to study. So आपके लिए ये जानना बहुत ज़रूरी है कि हम पूरे एंजियोस्पर्मिक प्लांट या फ्लावरिंग प्लांट का जो फ्लावर है उसमें एग्जैक्टली कहाँ पे स्टडी कर रहे हैं. So that's the point where we need to discuss. See here. So this is stamen that I have already discussed. पिछले वाले स्लाइड पे मैंने फ्लावर का वो स्ट्रक्चर बताया जिस पे आज हमारा डिस्कशन होने वाला है. तो हमारा डिस्कशन इस पार्ट पे है, स्टेमन पार्ट पे. स्टेमन के मेजरली दो पार्ट होते हैं. एक जो आपको टर्मिनली दिख रहा है, एक बाइलोब स्ट्रक्चर दिख रहा होगा, एक पहला लोब और एक जो उसकी छोटा सा जो पीछे की तरफ आपको नजर आ रहा है वो सेकंड लोब है. सो टर्मिनली आपको एक बाइलोब स्ट्रक्चर दिख रहा है और उसे हम लोग क्या नाम देते हैं? उसे हम लोग नाम देते हैं एंथर. और ये जो लॉन्ग स्लेंडर पार्ट, ये स्टॉक जैसा पार्ट आपको नजर आ रहा है, व्हाट इज दैट? दिस इज फिलामेंट, दिस इज फिलामेंट. सो anther and filament makes the stamen, makes the male reproductive organ of a flower. Very important because male reproductive organ में ही बनेंगे male gametes. Yes. So let me tell you students, this is a structure of a typical, this is a structure of typical stamen. And here तो हम जो बात कर रहे थे, रिमेंबर कि मेल गैमेट्स कहाँ बनेंगे? पोलन ग्रेन्स में. तो वो पोलन ग्रेन्स एक्चुअली फ्लावर के अंदर, स्टेमन के अंदर कहाँ बनते हैं? दे आर प्रोड्यूस्ड इन एंथर पोर्शन. तो यू आर स्मार्ट इनफ टू अंडरस्टैंड कि अब अगर हमें मेल गैमेट के फॉर्मेशन को समझना है तो हमें अपने स्टडी को किस तरफ डायरेक्ट करना होगा? सो वी नीड टू स्टडी anther. There only we will see formation of pollen grains, where ultimately at one point of time what will be formed? Male gametes. So our next point or our next objective will be to understand what is there inside a anther. See here. So this is how your anther looks. This is the TS of the anther. Very, very important. Okay. So I hope you people are trying to understand that what flow I am using. Our target is to reach a point to understand where exactly male gametes are produced. Keep this thing in your mind. So what is this? This is TS of anther and it is cut like this. Okay, you hold anther like this and just try to slice it from the top. So this is how it will look. So when you want to ponder, जब आप ऊपर से देखेंगे इसको एंथर को तो आपको ऊपर से क्या नजर आएगा? आपको ऊपर से एक ऐसा स्ट्रक्चर नजर आएगा, राइट? इसमें बीच का जो पोर्शन है उसे कनेक्टिव कहते हैं. ओके, आई नीड टू राइट इट अगेन फॉर यू. इसे कनेक्टिव कहते हैं. एंड व्हाट इज द फंक्शन ऑफ कनेक्टिव स्टूडेंट्स? इट इज अ स्टेराइल टिश्यू. इट हैज नथिंग टू डू विद द रिप्रोडक्शन पार्ट. और कनेक्टिव के दोनों तरफ, राइट, एक इस तरफ और इस तरफ, बोथ द साइड ऑफ द कनेक्टिव, राइट एंड लेफ्ट, देयर आर टू lobes and each lobe possess two sacs. See here students, ये एक लोब है. अगर हम कनेक्टिव से एक लाइन ड्रॉ करें तो here you can see one lobe and here you can see two lobes. So you can see what? Anther is actually bilobe. So टिपिकल एंथर, यह एक टिपिकल एंजियोस्पर्मिक प्लांट का एंथर कैसा दिखता है? बाइलोब्ड होता है. कैसा होता है? बाइलोब्ड होता है. टू लोब्स होते हैं कनेक्टिव के दोनों तरफ. नेक्स्ट इज डाइथिकस होता है. व्हाट इज एंथर? व्हेन आई से a typical anther is bilobed, that means presence of two lobes. When I say an anther is dithecous, that means each and every lobe of anther is going to possess two sacs or two thecas. See here very carefully. So this is what? One lobe. So each lobe is possessing what? Two thecas, right? Two thecas. Here also two theca, right? And that is why anther is known as dithecous. A typical anther is known as dithecous because each and every lobe of the anther possess two thecas, right, or two sac-like structures. Second point is a typical anther possess 4 microsporangia. A typical anther possess 4 microsporangia. What is microsporangia? Microsporangia is actually a sac-like structure, and when anther is going to mature, this microsporangia will convert into pollen sac when pollen grains will be formed inside a mature anther. That's the future of what? Microsporangia. Please remember this. So students, what you understood is in a typical anther overall how many thecas are there? Overall 4 thecas are there, and these 4 thecas are known as microsporangia, right? And a single will known as microsporangium. So in all or overall how many microsporangiums or how many thecas are there? There are total 4, and that is why we call a typical anther as tetrasporangiate. Therefore, we call a typical anther as tetrasporangiate. Why tetrasporangiate? Because of the presence of 4 microsporangia. 4 theca-like structures inside a typical anther. And as you can say, these thecas are present at the 4 ends, hence giving anther a tetragonal shape, 4-sided, right? I hope this much is clear to everyone. Now students, what we got to do next is, see, हम लोग कहाँ से आ गए, हम लोग फ्लावर पे थे, राइट? फ्लावर से हम स्टेमन पे आए, स्टेमन से हम एंथर के अंदर गए. अब एंथर के से नेक्स्ट what we got to study inside an anther is the structure of a single theca or single microsporangium. Microsporangium is the place where pollen grains will be formed in future, and at that point of time you will call that particular microsporangia or microsporangium as pollen sac. So, in there exactly, as you know, in pollen grain male gametes will be formed. So, our next target is to understand how does a microsporangium look like. So, now अब हम लोग और ज़्यादा डिटेल के अंदर जा रहे हैं एंथर की. So, अब नेक्स्ट स्लाइड पे what are we going to study is, नेक्स्ट स्लाइड पे we will study how exactly this structure look like. Okay. Okay. So, this is what now our whole concern is to understand this much portion, right? This much portion where I'm going to teach you two things: how exactly a microsporangium from inside look alike, and what are the layers or walls that surrounds a single microsporangium. So, let us see students, let us see that what are the 4 types of walls that surrounds a microsporangium as well as how it looks. So, a microsporangium appears nearly circular in outline. You can see this. These are microsporangium, and each microsporangium is surrounded by 4 layers of anther. Now, let us see what are those 4 types of layers. See here students, I am trying to cover this much portion. Okay, although when I am going to talk about layers, that those layers are all obviously, that means these 4 layers are going to cover the entire anther. They are not only confined to a single microsporangium. Okay, but what I am trying to say here is next time in the next slide where I am going to show you how microspores are produced, our main focus will be this area. So, मैं आपको एडवांस में ही बता रही हूँ कि अभी हम लोग कहाँ से स्टार्ट किए थे, स्टेमन से स्टार्ट किए थे, और अब हम लोग कहाँ तक आ गए हैं, हम एंथर के अंदर इनर स्ट्रक्चर तक आ चुके हैं, और वॉल्स के बाद सीधा जो हमारा नेक्स्ट टारगेट होगा वो होगा इसे समझने का. आई होप दिस मच इज क्लियर. सो सी स्टूडेंट्स, सो एंथर का जो सबसे बाहर वाला स्ट्रक्चर है या सबसे बाहर वाली जो लेयर है, द आउटर मोस्ट लेयर ऑफ द एंथर, इफ यू गो फ्रॉम आउटसाइड टू इनसाइड, इज एपिडर्मिस. Epidermis. And epidermis is generally single cell, a wall which is made up of single cells. Okay. Next the inner is layer. Next to this layer is this layer, right? This wall or this layer, this is made up of what is the name? The name is endothecium. Endothecium, right? First is epidermis, inner to epidermis is endothecium, then inner to endothecium are middle layers. Middle layers. Yes students, I am using this word layer because they are generally 2 to 3 layered. This wall is generally 2 to 3 layered. Okay. And the innermost layer that is actually directly surrounding the microsporangium. What is microsporangium? This space. This this bag-like structure is known as microsporangium, and this is the layer that is directly surrounding the microsporangium, right? And it is known as tapetum. Okay, right? So, epidermis is single layered wall, endothecium is also single layered wall, middle layer can be 2 to 3 layered thick, and tapetum is again single layer. So, these are the 4 types of layers that is surrounding a microsporangium inside a anther, right? See here students. Now, let me tell you in short what is the function of these layers. So, there is a very beautiful line in NCERT that you need to understand, that what NCERT says that epidermis, endothecium and middle layers, they collectively perform 2 functions, right? One is the protection of the internal structure of the anther. Obviously that means microsporangium and the structures that are present inside it, right? And along with protection, what is their function? Their function is dehiscence. What is dehiscence? Dehiscence means when the anther is mature, it is filled with pollen grains at that point of time. Okay, listen, understand it like this. At the time of maturity of the anther, pollen grains will be present inside it. And what is the objective of the pollen grain? To come out of the anther and ultimately try to reach the stigma of the female part, right? So, see here, at the time of maturity, the walls of the anther will crack and the pollen grains will come out. So, this process where walls of the anther cracks, right, and the pollen grains comes out is known as what? Dehiscence. Yes, it has a separate mechanism that we can discuss in another lecture in detail, but right now what you should understand is the outer three layers, epidermis, endothelium and middle layer, have a collective function of protection, giving protection to the anther and the inner parts of it, as well as they helps in dehiscence of the anther, that means rupturing of the walls and coming out of the pollen grains. So, their function is protection, collective function. They also have their specific function, but as of now I am telling the collective function, protection and dehiscence, right. Now, let us talk about this tapetum. What is the function of the tapetum? Can you tell me, students, that what is the function of tapetum? Tapetum, actually it is nutritive in nature. So, it will provide what? Nutrition. It will provide nutrition to sporogenous tissues, or you can say microspores, or sporogenous tissues also you can write. See, students, why am I writing like this? Because ultimately the objective of tapetum is to provide nutrition. To provide nutrition to whom? To all those structures that are present inside it or inner it, not inside it exactly, I should say inner to this. So, this layer, if I say this is tapetum, so it will provide nutrition to all the cells that are surrounded, that tapetum itself surrounds it. Now, understand, initially when anther is young, the type of cells inside this microsporangium that are surrounded by tapetum are actually homogeneous, right. After some time they differentiate and become what? Sporogenous tissues. After some time they differentiate and become microspore mother cells. Then there is a division, reductional division that take place inside microspore mother cells, and what are formed? Haploid microspores. So, continuously the work of the tapetum is to produce or provide what? Nutrition to what all structures are formed inside or the structures that itself are being covered by the tapetum layer. I hope this much is clear to all of you, right. So, now let us see, we have reached from outside of the stamen to the microsporangium, right. Now, our objective is to understand where exactly pollen grains will be formed. Yes. So, please remember, in the beginning of the session I told you male gametes will be produced where? They will be produced ultimately in pollen grain, right. So, अगर आपको समझना है कि मेल गैमेट कहाँ बनेगा, किस जगह पे बनेगा, तो उसके लिए आपको क्या समझने की जरूरत है कि पोलन ग्रेन कहाँ और किस तरीके से बन रहा है. Yes, so now we are at the point where we can understand how and where pollen grains will be produced. So obviously till now you should be clear कि pollen grain कहाँ बनने वाला है. pollen grain उस microsporangium वाले portion में बनने वाला है. जब pollen grain बनेंगे तो आप उसी section को pollen sac भी कह सकते हैं, right? So this whole process, in this whole flow chart, beautifully made flow chart, what I am going to do is I am going to explain how exactly pollen grains will be formed, right? And where pollen grains will be formed, that I have already covered in the previous session. Here exactly we are talking about. So अब तक आपको प्लेस क्लियर हो जाना चाहिए कि ये पूरा जो काम हो रहा है पोलन ग्रेन बनने का, मेल गैमेट बनने का, ये सारा जो स्ट्रक्चर, ये सारा जो काम चल रहा है वो कहाँ चल रहा है? यहाँ, right? So now let us see how pollen grains will be formed, right? And not to forget, inside pollen grain at maturity at certain point of time in angiospermic plants male gametes will be produced. Also it also has a variance of 40 to 60%, but that we will discover or I will discuss in the next session, right. So as of now what you can keep in mind is here we will understand where exactly pollen grains or how exactly pollen grains will be produced. So see, the process is known as microsporogenesis. So the objective of this whole discussion is to understand how microspores will be produced, and not to forget from microspores only pollen grains are produced. So अगर आपको pollen grain कैसे बनता है ये समझना है तो उससे पहले आपको microspores कैसे बनते हैं ये समझना बेहद जरूरी है. तो हमारा सबसे पहला काम क्या है? टू अंडरस्टैंड हाउ microspores are produced inside that microsporangium. So let us start. The heading is microsporogenesis, and what does microsporogenesis mean, students? Microsporogenesis is a process that is taking place inside an anther where microspores are produced from a diploid microspore mother cell. So see how it takes place. So obviously the place should be clear, this whole process is taking place inside microsporangium, right? So how it starts? It starts from this stage when anther is young, right? Inside a microsporangium, what will happen? When anther is young, inside a microsporangium, the tissues or the cell, these cells are actually homogeneous or meristematic. They are not differentiated. After some time, what will happen? They will differentiate, okay? They will become sporogenous tissues. Then what will happen? Further differentiation will take place, and ultimately these cells will be termed as or named as microspore mother cell, right? Now please do not forget that anther is a part of sporogenous, or I can say anther is a part of sporophytic plant body, and each and every cell of sporophytic plant body should be diploid. So all these, if I talk about the ploidy, all these cells are diploid in nature. Not to forget this. बहुत फ्रीक्वेंटली आपसे प्लॉइडी के बारे में क्वेश्चंस पूछे जाते हैं. तो माइक्रोस्पोर मदर सेल विल ऑल्सो बी व्हाट? इट विल ऑल्सो बी डिप्लॉयड टू एन, राइट? नाउ ultimately in microspore mother cell what will happen? Reductional division will take place, meiosis will take place, and ultimately what will form? 4 haploid microspores will be formed. 4 haploid microspores will be formed. But initially all those 4, because it is meiosis, obviously the ploidy will change from 2n, the cells will become n, right? So not to forget that initially all the 4 microspores will be connected to each other with the help of a callose layer, right? But after some time, at the time of maturity, with the help of callase enzyme, what will happen? That wall of callose will be dissolved and the microspores will separate, right? And that time what you will call them as pollen grains, right? Now this pollen grain will ultimately after several changes will convert into what? Male gametophyte, and in this male gametophyte what will be produced? Male gametes. Yes students, तो यहीं से हमने हमारा ये पूरा जर्नी स्टार्ट किया था जब मैंने आपको ये कहा था कि आज के इस सेशन में हम लोग ये समझेंगे कि कहाँ पे और किस तरीके से मेल गैमेट बनेंगे एक एंजियोस्पर्मिक प्लांट में, एक फ्लावरिंग प्लांट के अंदर. सो सी हियर वी स्टार्टेड फ्रॉम स्टेमन, right, and we have reached to microsporangium, and in a microsporangium we understood that how using microsporogenesis a male gametophyte will be produced. That's the whole process that I wanted you to understand, right? So after several changes, it's not students के एकदम से मतलब माइक्रोस्पोर्स बने और माइक्रोस्पोर से एकदम से ऑल ऑफ अ सडन मेल गैमेट्स नहीं बनते. देयर विल बी सर्टन डिविजन और देयर विल बी सर्टन टाइम्स दैट यू विल सी दैट काफ़ी सारे चेंजेस होने के बाद इवन माइटोसिस भी होगा, उसके बाद में जाकर आपको क्या मिलेंगे? आपको मेल गैमेट मिलेंगे. नाउ टू बी प्रिसाइज, इफ़ यू से आई कैन टेल यू स्टूडेंट्स, ये वाला पोर्शन ये वाला पोर्शन जिनके लिए भी क्लियर नहीं है, मैं थोड़ा सा और इसको क्लियर करना चाहूँगी कि जब माइक्रोस्पोर्स बन जाते हैं, नाउ ऑल ऑफ यू नो दैट माइक्रोस्पोर्स इज हेप्लॉइड इन नेचर। जब माइक्रोस्पोर्स बन जाते हैं तो माइक्रोस्पोर का जो न्यूक्लियस है वो क्या करेगा, वो माइटोसिस शो करेगा और उससे दो सेल्स बनेंगी, एक जनरेटिव सेल और एक ट्यूब सेल, राइट। तो इस स्टेज में जब एक माइक्रोस्पोर के अंदर जनरेटिव सेल भी आ जाता है, वेजिटेटिव सेल भी आ जाता है और उसके ऊपर स्पोरोपोलेनिन की लेयर बन जाती है, इनसाइड इंटाइन की लेयर होती है और बाहर स्पोरोपोलेनिन की लेयर बन जाती है, तब ऐसे एक स्ट्रक्चर को आप क्या कहते हैं, पोलन ग्रेन कहते हैं। और पोलन ग्रेन को ही फर्स्ट सेल ऑफ मेल गैमेटोफाइट कहा जाता है। फिर इसके अंदर भी सर्टेन चेंजेस होते हैं और फाइनली एक मेल गैमेटोफाइट का फॉर्मेशन होता है, जिसके अंदर जाकर फिर से माइटोसिस डिवीजन करने पर जनरेटिव सेल में फिर से जब माइटोसिस होते हैं तब जाके आपको फाइनली मेल गैमेट मिलते हैं। बट नॉट टू गेट कंफ्यूज, ये जो लास्ट में आपको मैंने पोर्शन समझाया है दैट आई विल डेफिनेटली कवर इन माय नेक्स्ट क्लास। आज का ऑब्जेक्टिव बस इतना समझाना था कि कहाँ और किस तरीके से पोलन ग्रेन का फॉर्मेशन हो जाता है। आई होप यू एंजॉयड द सेशन टुडे स्टूडेंट्स, दैट्स ऑल व्हाट आई वांटेड टू एक्सप्लेन till now. Okay, if you like the session, please give me a thumbs up and do subscribe to Infinity Learn by Sri Chaitanya. Do not forget to give me comments, that will help me to improve my next lecture. Thank you students for staying connected.